Hi everyone, welcome to this session on the topic of discovering the trees of life from genomes to phylogenies. I'm chairing this session with Rebecca Stubbs. This is the second session. We had our first session yesterday, and we have a range. We have a list of fantastic speakers talking about fantastic topics. So you know how if you attended previous sessions, you know that first I'm going to introduce the speakers and their talks. Then you can type your questions in the Slido just under the stream. Please type the name of the speaker before your question, so at the end we know who is the target of your question. So we'll see all the talks one after each other, the pre-recorded videos, and then at the end we will sort out your questions. All right. Remember that at the end of the event, so we'll finish at 10 to 7 UK time. At the end of the event, you can join the networking room in the special app that you can find in the networking section in the left menu. All right. So I'm going to introduce the speakers. All right. So first we have Leticia de Paula, who will talk about phylogenomic analysis of Tachinidae. Then we have Jonas Lesquart, who will talk about complex whole genome phylogeny of elusive neotropical small cats. Then we have Silvia Perea, who unfortunately, or maybe fortunately, she cannot be here because she's doing fieldwork. So she told us that if you have any question about her talk, please send her an email. And Silvia's talk is about selective constraints on phylogeny in the visual pigments of a cavefish. Then we have Tom Williams, who will talk about the phylogenetics and the early evolution of bacteria. And finally, we have Heike Björk-Drost, who will talk about Diamond, so one of my favorite programs. So the title of the talk is Sensitive Protein Alignments at Tree of Life Scale using Diamond. So without further ado, we are going to now start listening to the talks.
Hello, my name is Leticia. I'm from the University of São Paulo, Brazil. I'm here to present my master's research that's soon going to be a paper with other collaborators, and it's phylogenomic analysis of Tachinidae, a transcriptomic approach to understand the subfamilies' relationships. So you guys can understand a little bit about the family. Tachinidae family is one of the biggest ones within Diptera. They have 8,500 described species. The nootropical fauna is the most diverse and the least studied. They are considered parasite of arthropods, and they are known to be biological control because they regulate populations of large groups. So the Tachinidae family is known to be divided into 4 subfamilies. So we have Exoristinae, Tachininae, Phasinae, and Dexinae. Those are the 4 valid subfamilies until now. Those are the most important studies, most important phylogenies for this group. We have in the left side the phylogeny I just showed you, which is a morphological phylogeny for the family. In the right side, we have a molecular phylogeny of the family also. Those are important because they have a deep sampling, and they also they're focused on the whole family. So they have a really similar results. They have Dexinae, sister to Phasinae, and Tachininae, sister to Exoristinae. Those are 2 clades formed by 2 subfamilies each. And they also recovered a 5th lineage, a 5th basal lineage, which is sister to the remaining subfamilies. They both have this basal lineage here. Based on everything I just showed you, our focus is to reconstruct a hypothesis of phylogenetic relationships of the family, the Tachinidae family, focused on the limits and relationships between the subfamilies. We want to establish the monopoly of the subfamilies. We want to verify the relationships between them and establish the sister group of Tachinidae. We used transcriptomic data, so we could recover a little bit more than 1,300 single copy protein coding genes. and this is one of our phylogenies. We have Pollenidae, a sister group for Tachinidae, which is really commonly recovered in the literature because most of the works that sample Pollenidae, they recover this family as sister group to Tachinidae. So here we established that with a high support. We also have the relationships for the subfamilies are the same that I just showed you in the two phylogenies. We have here Dexine, sister to Phasine, and Tachinine, sister to Exoristine. We have those two clades who composed with two subfamilies each. And this is recovered with a high node support also. And we also recovered the fifth lineage. We have this fifth lineage also recovered with high node support. We covered the Myophazine tribe, member of the fifth lineage. So here we establish the existence of the fifth lineage and that Myophazine is a part of it, because every work that sampled Myophazine, they recovered this tribe within the fifth lineage. Here we established that with high node support. And we cannot establish a fifth subfamily because for that we would need more sampling for Myophazine and more sampling for the other tribes that are possibly members of the fifth lineage. So here we just raised the questions about if Tachinidae really have only 4 subfamilies, but this for the fifth subfamily to be established, we would need a deep study for this. Okay. We covered Phasine as monophyletic, which is really commonly recovered in the literature. We established that high node support also. We have Dexine as monophyletic too. This subfamily sometimes recovered as paraphyletic, but here recovered as monophyletic with really high node support. We have Exoristine as monophyletic too, which is commonly recovered as well in the literature, and here with high node support too. And finally, we have Tachinine as polyphyletic. This is also commonly recovered in the literature because Tachinine is the one subfamily that most of the times is recovered as a not monophyletic subfamily. So here we have the fifth subfamily that I just talked about. We have the main Tachinine clade, and we also have here within Exoristine the tribe Iseline. This is important because Iseline is a neotropical tribe that was previously recovered within other subfamilies and only based on morphological similarities. Here is the first work, our study is the first one to sample the Iseline tribe for a phylogenetic study. So this is important because we question the position of Iseline based on phylogenetic criteria. So this is important, we raise these questions, but we cannot again establish anything because Iseline is a poorly studied tribe. We would need a morphological revision and more sampling for this tribe with more genera and more species to really establish the position of this tribe. So here we present the first phylogeny of Tachinidae using transcriptomic data. We have the Tachinidae family as monophyletic. We have Pollenidae established as sister group for Tachinidae. We also establish the relationships for the subfamilies. We have 3 out of 4 subfamilies as monophyletic. We have Tachinine as polyphyletic, and we established the 5th lineage with Myophazine as part of it. So I really would like to thank the opportunity to present my work and the attention of everybody. Thank you very much.
Hi, my name is Jonas, and I'll be talking about the rather complex phylogeny of the cat genus Leopardus. So Leopardus is a genus of about 10 species of neotropical small cats. And my colleague Fernanda recently published a new phylogeny for this genus based on SNP array data. And within this phylogeny, we can see a deep paraphyletic situation for the Tigrina group, which you can see here in red, one species, in orange, another. And this purple sample, which should be part of the orange clade, is separated from those Tigrinas by two completely different species that actually have quite a distinct morphology from the basic Tigrina morphology. So you can hear more about that in her poster talk in this conference, or scan this QR code to go to the paper. And within this phylogeny, two key locations of the Tigrina group are still unsampled, as you can see here. And also one species is missing, the exceedingly rare Andean mountain cat. There's a lot of taxonomic disagreement within this genus, and part of the cause of that is the extensive phylogenetic discordance that we observe, which is caused by both incomplete lineage sorting and hybridization. And a previous study used 4 complete genomes of Leopardus species, and they found 3 topologies to be more or less an equal frequency spread across the genome. So they used various partitions here, but there's not really one topology significantly more frequent than the others that you can just point at and go, oh, this topology reflects the true speciation history. So we want to further address this with whole genome sequencing of all the species of Leopardus, which brings us to the current data set composed of 16 genomes covering all 8 species recognized by the IUCN Cat Specialist Group. And we use short-read data at coverage depths of about 17 to 27 fold. And as a reference genome, we use the Geoffrey cat assembly made by collaborators in Texas, as well as the outgroup species Canada lynx assembly from the Vertebrate Genome Project. Going to the first results, we see the pairwise sequence divergence. So we chopped up the genome in separate fragments of 100 kb length each, and from each of those fragments, we construct a distance matrix. And when we look at the average, the genome-wide average of those matrices, we find that there's a sequence divergence among species of Leopardus of about 0.86% and up to almost 0.5% within single species, reflecting its recent speciation history. From those 21,000 distance matrices, we then made a neighbor joining consensus tree, which you can see here with bootstrap support as well as node frequencies indicated. And the high bootstrap support indicates that we reliably retrieve the same consensus tree when we subsample the matrices with replacement, whereas the individual node frequencies actually point to quite a lot of discordance going on along the genome. And in the tree, you can see that we sampled this group with Colombian samples, and that it clusters with the Central American Tigrina, reinforcing this deep paraphily. We did the same on those 100 kb fragments with maximum likelihood trees rather than neighbor joining trees. Here you see all of them overlaying on top of one another, with the gray areas reflecting the discordant positions. And this is a majority rule consensus tree, again with node frequencies. And it's very similar to the previous tree. This is another consensus representation of the maximum likelihood trees. It's a network configuration just to highlight some of the most prevalent crosslinks within the tree. And on this consensus tree, we are showing in bold something called the leaf stability index, which is a measure of how mobile a certain taxa is throughout the set of trees that we have. And lower is more mobile, and we see that the most unstable taxa are the margay here and the Andean mountain cat. Now, if you see the Andean mountain cat attached to the margay and only 45% of the genome really, then the natural question to ask is, where else does it attach throughout the genome? And we counted this with the branch attachment frequency shown here in red. And you can see that in 19% of the genome, the Andean mountain cat attaches to the ocelot rather than to the margay, and so forth. Now zooming in a bit on the Andean mountain cat, because it's the only species of Leopardus that is listed as endangered on the IUCN Red list. It has quite a special ecology. It lives high up in the Andean mountains, and it has a small population size of about 1,400 mature individuals during the last census. So we looked at its genetic diversity, and we found that we measured this as autosomal heterozygosity, broken into 200 kb windows and given as a box plot here, and we see that it's very low compared to other species of Leopardus. Here you see the ocelot with the highest genetic diversity in Leopardus, and the Geoffroy's cat here reflects sort of the average genetic diversity. And to exclude recent inbreeding as a cause for this so low genetic diversity in the Andean mountain cats, we looked for runs of homozygosity throughout the genome, but really we found only one large run of homozygosity here shown on the B2 chromosome, and then some very tiny ones, altogether constituting not even 1% of the genome in runs of homozygosity. And the absence of rows combined with the low genetic diversity is suggestive of a long-term small population size for the species. Then going on to what usually is a conclusion slide, but here it's a next step slide, because this is all very much preliminary data. So we plan to do introgression tests to distinguish between ILS and hybridization as root causes of the discordance that we observe. And then to track that discordance through the genome, we want to correlate local topologies with genomic features like recombination rate, and to fill a few sampling gaps, especially the Guianas here. We want to sequence museum specimens. These are the references, and I want to thank my PhD supervisors, Hannes and Eduardo, as well as the various funding bodies, all the collaborators. And you can reach out to me with this email and find the project on ResearchGate as well. Thank you for watching.
Good morning. I'm going to introduce here this interesting freshwater fish that is Asthenas mexicanus, which is conformed by two different contrasting morphotypes related to the adaptation of cave life. Well, the genus Asthenas is the most diversified genus of the family Carassidae, with more than 140 species, and it has an Arctic distribution. Within the genus, Asthenas mexicanus is the type species, and it is distributed in North America from Texas and New Mexico in the north to the eastern and central part of Mexico in the south. And as I have told before, there are two different morphotypes within the species. A surface form at the top of the bottom, at the top of the slide, which has a well-developed eye and this characteristic coloration, and a cave form, which is in the top of this slide and has, in this case, the completely loss of eyes and loss of pigmentation, although there are different levels of eye development with this form, troglodytic forms. In this study, we have focused on the visual systems, and the visual pigments are the molecules implicated in the process of the vision. Vision is a communication system that allows organisms to interact with their environments. And these visual pigments are constituted by a transmembrane protein, the opsin, with a chromophore, which is the molecule that makes the phototransduction process mechanism. Within the visual pigments, within the opsins, there are different kinds of these molecules. The visual ones are the ones expressed in the eye retina and mediate visual perceptions, and are constituted by the Silvari photoreceptors sensitive to different length waves of the light spectrum and the rod photoreceptors, the rhodopsins implicated in the dim light visions. But there are also different non-visual opsins with different functions and expressed in different tissues. Regarding the phylogeny of Astianus mexicanus, the case, most of the studies have are congruent in that cave populations don't constitute a monophyletic group, and at least 2 different events of colonization have occurred. Here I present you a recent phylogeny based on whole genome sequencing, which is interesting because it demonstrated that divergence between cave and surface population was younger than previous estimates, and that there are gene flow among cave and surface populations. And in spite, there is this gene flow, these phenotypic traits related to troglomorphic forms have been kept in the evolution. In this study, we try to resolve 2 different evolutionary questions. On one side, it is expected that vision may confer a selective advantage for life in dark conditions. Therefore, how selection acting in this process? And on the other hand, we want to try to resolve if the evolution of visual pigments in Astianus cave fish is in concordance with the general evolutionary history of the groups. Therefore, we evaluate the action of selective pressures on visual pigments in Astianus mexicanus and reconstruct the phylogeny of this complex based on the visual pigments. At the moment, we have analyzed 16 cave and 4 surface populations geographically distributed in 3 mountain ranges in the Sierra de Madre Oriental, in which both old lineage and new lineage are distributed. And we have analyzed 2 individuals per populations. For these individuals, we have obtained sequencing, whole genome sequencing, and we have used the genome reference of Astianus mexicanus, published. And we have extracted 7 visual opsins and 31 non-visual opsins for these organisms. And finally, we have performed selections analysis in order to estimate the omega value, the coefficient of selection, and to reconstruct the phylogenetic tree. At the moment, we have preliminary results just based on these 7 visual opsins. If we can see here in the table, there is a general pattern of predominance of negative or pre-fine selection, as all of the omega values estimated were lower than 1. And we have also seen that these omega values were always higher in cave populations than in surface populations, which suggests a relaxation of selective constraints in cave populations, which is strong in the old lineage populations than in the new lineage population caves. These results are in agreement with previous studies in other freshwater cave species, such as the case of the Cynocyclocaelus genus Asiatic and the cave species of the Amblyopsidae family. We also identify several deletions that could be potential loss of function mutations, such as premature stop codons or mutations that change the protein reading frame. These loss of function mutations are also found in other freshwater cave species. However, the diversification of these other species is older than that estimated from Astianus mexicanus. For this reason, we have to be careful when extracting robust conclusions, and we need to do more empirical, to obtain more empirical evidence. And finally, we also reconstructed the phylogeny of the Astianus mexicanus based on these visual opsins, and we were able to recover the old lineage and the new lineage identified for Astianus mexicanus. As we can see here in the phylogenetic tree, in the old lineage, the cave populations are differentiated from in red, are differentiated from the surface populations in blue, which is not the case for the new lineage, probably as different evolutionary time in both. Therefore, the diversification of visual obscenes in Ascinas mexicanus complex is congruent with the evolutionary group. Finally, next questions that we have to resolve is what is the pattern, general pattern that we can find in the non-visual obscenes? Is similar to that find in visual obscenes or are different? What happened with other genes that are related also with the vision process, but that are not obscenes? Or what happened with these loss of function mutations? Are they real or are they assembling artifacts? All these questions will help us to understand a new piece of the interesting puzzle that constitute the evolution of cave organisms. And that's all. Thank you for your attention. If you have any questions, you can always write to my mail or to contact with my research group. Thank you.
So hi everyone, and thanks for coming to listen to my talk. Today I'm going to tell you about some work that I and some collaborators have been doing on the early evolution of bacteria, the deep phylogeny of bacteria. And I'll be showing you a couple of the main results from a recent paper of ours, which has been published. It's Coleman et al. 2021. So if you're interested, you can check that out after the talk. And I wanted to say that this work has been very collaborative between my group and also the groups of Anja Spang at NIAS in the Netherlands, Gergely Saloshi in Budapest, and Phil Hugenholtz in Brisbane. So let's see. I'm going to start by sort of motivating why we want to study bacterial evolution. I mean, it's a fascinating topic, right? And bacteria are among the most diverse, genetically and metabolically diverse cellular life forms on the planet today. And as far as we can tell, that's been true throughout Earth history as well. The planet we live on today has been totally altered by bacterial activity. The fact that we breathe oxygen, of course, has got to do with the evolution of cyanobacteria. And recently we've discovered, the community has discovered many new groups of bacteria, sort of an enormous diversity. One of the sort of an iconic, I guess, image from a relatively recent paper shows the diversity of life as revealed by environmental sequencing. And the impression you get from this tree from Hug et al. In 2016 is that most groups of life are prokaryotic, and maybe much of that diversity is bacterial. So there remain huge open questions, very basic questions about the nature of the first bacteria and how early bacterial evolution proceeded. And in this talk, I'm going to talk about really two of those. First, is there a tree of bacteria? Some people have argued that there's so much horizontal gene transfer in bacterial evolution that this tree structure is kind of a misleading way to think about bacteria. If there is one, where's the root of that tree? And what was the last bacterial ancestor like? And I'm going to be talking about three major groups, or candidate groups, if you like, throughout the talk. The first is the Pterobacteria. That's the name of a group that includes Firmicutes, Actinobacteria, Cyanobacteria, and many other lineages. The Gracilicutes, so those include the classical diderm bacteria, so the gram-negative bacteria, things like Proteobacteria, Chlamydia, Bacteroidetes, and groups like that. And also a relatively recently discovered group, the candidate Phyloradiation. So they have small cells and genomes. And in that tree I showed you on the previous slide, it looked like they might represent one of the deepest splits in the bacterial tree. So one of the problems with trying to root the tree of bacteria is that the normal strategy for rooting trees, outgroup rooting, doesn't work very well. So the reason for that is that if you include the closest outgroup to bacteria, the archaea, then the longest branch in the evolutionary tree you infer is the one between the two domains. And that can interfere with the resolution of the within bacteria relationships due to long branch attraction, a phylogenetic artifact. The other issue is that if many bacterial and archaeal genomes have sort of 2,000, 3,000, 4,000 genes on them, there's only really 30 or 40 genes that seem to evolve more or less vertically across archaea and bacteria that you can use to reconstruct that tree. So you're having to set aside a lot of the genomic data we might have to resolve the relationships. So if we could root the tree without an outgroup, that might bring some new insights. There's different ways you can do this, but in this study we used a method called gene tree species tree reconciliation. So the basic idea there is that you compare gene trees of every gene family or many gene families to a species tree, and you use the differences between them to try to work out the history of gene duplications, transfers, and losses within each gene family. And if we use a probabilistic model for gene tree, species tree reconciliation, so a model where the individual gene trees evolve along the species tree and occasionally experience events like gene transfer between lineages, for example, we can use that model to test the probability of different roots, different species tree roots, if you like, given the data. And the biological intuition of what's happening here, I guess you could think of this as an extension of the classical method that's been used to root the whole tree of life using gene duplications, right? So if you think about the classic studies of Iwabe or Gogarten, they used anciently duplicated genes to show that bacteria seem to be the deepest split in the tree of life. We can look at all of the duplications and also transfers and losses in bacterial evolution to try to root the bacterial tree. And when we did that, so we applied this method to 11,272 gene families from 265 bacterial genomes. The result we got was that the tree was rooted between the Gracilicutes and the Terrabacteria, with the CPR, so this recently discovered very diverse group, apparently grouping within the Terrabacteria as sister to the Chloroflexi. And when we took all of the gene trees for these 11,000 gene families and mapped them onto this species tree, we found that on average over 2 data sets, 66% of those gene tree branches mapped onto the species tree branches. So we think that this kind of rooted framework, if you like, provides a summary of an important component of bacterial evolution, a vertical component, notwithstanding the enormous impact, of course, of gene transfer as well. So this could be a vertical trace in bacterial evolution. No approach is perfect, of course, and more recently, so in some unpublished work, we've been looking at lots of other ways of rooting the bacterial tree. And here's one result. So for example, the MAD method, which uses branch length information, also roots the tree between Gracilicutes and Terrabacteria. You're on a much larger data set. There's been a number of other studies recently from different groups suggesting that relationship between CPR and Chloroflexi, and potentially some support as well for the Gracilicutes Terrabacteria root. So we wanted to use this rooted phylogenetic framework to try to infer something about the nature of the last bacterial common ancestor. And to do that, we took the COG ontology, so the functional classification of gene families, and we mapped COG gene families to the root, the inferred root of the tree. When we did that, we inferred that the last bacterial common ancestor had a relatively modest genome size, I guess, for a free living bacteria, so 1.6 to 2.7 megabases. It was difficult to resolve the ancestral metabolism because we found components of a number of different pathways, like the Wood-Lungdal pathway, for example, the reverse TCA cycle. But we think, given the presence of some components of the Wood-Lungdal pathway, and also the RNF complex may well have been an acetogen, which is in agreement with some hypotheses about the nature of early bacteria. Had some other interesting characteristics as well, like it was rod-shaped, had a flagella, a CRISPR-Cas system, and it seemed to have been a diderm or double membrane bacteria, suggesting that gram-positive bacteria, single membrane bacteria, may have secondarily lost the outer membrane. And if you're interested in this, there's been a number of studies looking at the question of the last bacterial common ancestor recently. Several of them support the idea that it was a diderm, but if you're interested, you should also check out Javier et al., who used a very different method for doing this, and came up with some similar and some different conclusions. So just to finish, it looks like from these analyses that the root of the bacteria is between 2 big clades, Gracilicutes and Terrabacteria, that CPR derived from within the Terrabacteria, and that early bacteria may have been acetogens and perhaps had a double membrane. So what we're doing now is we're working on better gene tree reconciliation, species tree reconciliation methods, also better substitution models for bacteria, and thinking about ways that we could put this rooted tree sort of embedded in geological time. So thank you, and future me will take your questions.
So it is the tree of life that brings us together here today. And it is the ambition of the Earth Biogenome Project to sequence and assemble up to 1.5 million eukaryotic species within the next 10 years. And so our group started to assess how we can use this comprehensive genomic resource to learn from the tree of life. And so it helps to remember, how do life scientists usually engage with the tree of life? And so what they do usually is they have their proteins or genes of interest. They would go to the basic local alignment search tool BLAST interface and would insert here their sequences, hit the BLAST button, and then hopefully retrieve some information from the NCBI non-redundant database, which reflects the tree of life scale data, genomic data nowadays. And so as a result, they would usually look at the top 100 or 500 hits, annotated in this list-like manner, where you know in which species, for example, the hit was found and what other type of annotation is available for this hit. And you can immediately see that if we have access to millions of genomes, looking only at the top 100 or 500 hits is really just a drop in the ocean. And so how can we actually retrieve way more information than this? And some of you may say, well, you know, we don't do this manually. Of course, we do heavy phylogenomic computations. And for that, of course, we use the command line tool of BLAST. And so we automate this entire process. But here I have to disappoint you a little bit, because we estimated that once we have access to millions of genomes and would want to do an all versus all comparison, BLAST will simply not scale anymore. So it may take years or even decades, depending on the resources that you have, to really traverse the tree of life in an all versus all comparison manner. And so the question is there a better tool out there, or some kind of future-proof tool for the Earth biogenome era? And so the answer is Benjamin Buchfink and Daniel Hewson at the University of Tübingen 6 years ago came up with a protein aligner called DIAMOND. And the ambition of DIAMOND was to exactly go into this microbiome field, and in particular metagenomics, and try to speed up the BLAST process to be able to traverse this treasure trove of genomic information, but at the cost of losing a lot of alignment sensitivity. But there basically speed was more important than sensitivity. But I was fortunate that Benjamin chose to join my newly established lab 2 years ago. And here I saw the opportunity to get inspired from this strategy, but now try to apply it to the eukaryotic world, and in particular, the Earth Biogenome Project. And so we tried to extend DIAMOND into version 2 and vastly extend it to be applicable to eukaryotic genomes, but trying to achieve the same sensitivity as BLAST, because of course, many people in the eukaryotic world and evolutionary or comparative genomics world care a lot about the alignment sensitivity. And so this is what we did this year. Here you can see a benchmark where you see on the x-axis the alignment sensitivity of the corresponding tool. Here the red dot is BLAST. And on the y-axis, you see the percentage speed up, or the x-fold speed up of the tool compared to BLAST. And what you see here is these two new sensitivity modes that we introduced into the new version of DIAMOND, namely the ultra sensitive mode and very sensitive mode, which now match the alignment sensitivity of BLAST while achieving 80 to 360-fold computational speed up. And even by losing a bit of sensitivity, we can still speed up the computation up to 8,000 times. And even alternative tools such as MM62 cannot achieve this, this speed and sensitivity at the same time. And so some of you may now say, well, you want us to apply DIAMOND to the tree of life. That's great. But which sensitivity mode should I use for the application that I have at hand? Because of course, there are many questions you can ask towards the tree of life. And the answer is that if you care about very similar proteins, let's say for functional annotation of your proteins, let's say 60% of sequence identity and above, using the fast mode is as good as BLAST, basically, but at the benefit of having 8,000-fold speed up. Whereas if you try to go more towards the twilight zone of protein evolution, for example, for deep homology questions or deep evolutionary questions, then we recommend using the very sensitive mode, which still matches the alignment sensitivity of BLAST. Whereas if you want to go beyond the twilight zone of protein evolution, then the only way to match the BLAST sensitivity is the ultra sensitive mode. But here I want to say that, of course, using a pairwise aligner for this kind of ultra deep homology assessment may not be the right choice in the first place. But just to give you an example of the power of this approach, we basically mimicked an experimental study, or we performed an experimental study to mimic this tree of life all versus all comparison. And what we did is we aligned the entire NCBI database, so NCBI non-redundant database, against the UniRef database. and computed roughly 32 billion pairwise alignments on a supercomputer hosted by the Max Planck Society. And here Klaus Reuter was really helping us a lot to scale DIAMOND in an HPC infrastructure. And what you can see here is that we align these 280 million versus 40 million sequences in less than 6 hours in the sensitive, very sensitive mode, and in less than 18 hours in the ultra sensitive mode. And just to give you an example of a comparison, so BLAST would use more than 2 months for the exact same computation. And it shows you that BLAST is simply not future-proof for the Earth Biogenome era. And so I hope I could convince you to at least have a look into DIAMOND and see if it can inspire your own application in this Earth Biogenome era. And I hope that together we can build this comparative biosphere genomics framework fueled by DIAMOND to really be able to learn from the tree of life within the next 10 years. And so Natasha Glower and colleagues have this wonderful figure that they have in their study where they try to show the applications of orthology to all these different comparative genomics methods. And I substituted this by adding here DIAMOND and trying to show you or hope that you can see that DIAMOND, or we intend to build DIAMOND as this core tool for exactly these kind of applications in the Earth Biogenome era. And so, of course, there are many people that helped us to make this possible, this entire research. And I would like to thank foremost Detlef Weigel, who is the director of our molecular biology department. And he really heavily supports our research. And then, of course, Benjamin Buchfink, who is the core developer of DIAMOND, and who really pushes very hard to make DIAMOND available now to the eukaryotic world and especially to the Earth Biogenome efforts, and many collaborators who inspire us and help us a lot to scale DIAMOND in various ways for various applications. And for this study in particular, Klaus Reuter, who helped a lot with the HPC infrastructure of DIAMOND. And I thank you for your attention. I hope I can answer some of your questions. Thank you very much.
And run through CD hit. I don't know how you guys say that.
Yeah, CD
okay,
hit.
Yeah, CD hit. Yes. So we took away the redundancies, the things that are the same. And after that, we choose the orthologs with Busco. And because we had so much orthologs, we used the e-value that Busco uses to say which alignment is the same, and they use the e-value. We use the e-value that they put in the table, that the output table. We use that to select which orthologs we're going to use. So we used only the e-values, the 0 e-values, and the e-values that are elevated to minus 100, and that is the least that we used. So we used we chose all the really good ones. So after that, we selected with bash scripts that we develop. Like, we're not bioinformatics, like we don't program, so we just make it work. We select that, and after that we aligned every gene, and we used IQ3 for the phylogenetic, the phylogeny that I presented. We also did TNT tree, and we are trying to do nucleotide tree because we used amino acids. I think that's it.
Fantastic, thank you very much.
Sure.
All right. We have two questions from Christina. Sorry, Christina, but Silvia is not here, so she cannot take your question. So you can send her an email. But she has one question for Heike. The question is, I am often interested to align proteins to genome, but in eukaryotes, this is quite challenging because of splicing. There are only old and slow tools for this task. Have you developed anything new about that in Diamond 2.0? Thank you for the answer.
Yeah, so excellent question. Unfortunately, we don't address particularly splice variants yet because we try to solve just traversing the tree of life for now, right? So the idea is to be fast and sensitive. So the only, hint I can give in terms of maybe what could work is to use this ultra-sensitive mode, because at least it is intended to be as good as BLAST while being still 80 times faster. So I guess this is the best we can do right now. But yeah, excellent question.
Thank you. And there is another one for you, but I think this one is brief. So is Diamond a tool to build phylogeny using whole genome resequencing data on a genus family level?
So no, it is not. But of course, any pairwise, all versus all pairwise alignment fuels a multiple sequence alignment construction and therefore phylogeny construction in general, right? So you can, it's basically the basic core tool that allows you to do quickly this all versus all comparison. So in a way, it is the basis for phylogeny. Yeah.
Thank you very much. All right, I have one question for Tom Williams. So that was a very cool talk reconstructing one of the oldest nodes in the tree of life. Do you think your reconstruction of the last common ancestor of bacteria is the same as what you would expect from Luca, or you really need to take into account the last common ancestor of Archaea and eukaryotes?
It's a really interesting question. I mean, I think some of these questions in sort of so-called deep phylogeny are really uncertain and really open. So I guess the consensus view is that the root is between bacteria and Archaea. And so if we want to reconstruct Luca, we ought to indeed account for both the bacterial common ancestor and the last archaeal common ancestor, and then work back from there. And although, there are interesting analyses that have suggested that Archaea might be within bacteria, we've been looking at this a bit, and the analyses we've done so far are consistent with that consensus root between Archaea and bacteria. But we know that that's a very difficult phylogenetic problem. It's a really tricky node to recover. And the consensus view is based on limited data, a few ancient gene duplications. So, now that we have all of this new data from all of these new lineages of Archaea and bacteria, who's to say what people are going to find when they look into that with new methods and so on? But there's a lot of work to do there. At the moment, I would say that those things would be distinct. And in particular, the idea that the last universal common ancestor might have been, a double membrane or something, this seems unlikely based on the distribution of that character across both groups of prokaryotes.
That's a very interesting proposition. So yeah, looking forward to see more of that. Okay, I have one question for Jonas. So I know that you mentioned that you are planning to look into introgression and ILS, but do you expect those factors to have a huge impact in the phylogeny you have reconstructed, or do you think it's going to impact only a few of the markers or genomic regions you use in your phylogeny?
No, we expect it to have a huge impact. So we find a lot of discordance, which I expect to be mostly explained by incomplete lineage sorting, but we also have evidence for both past and ongoing hybridization between multiple pairs of species within Leopardus. So yeah, I expect to find both. And the question is how much each of them contributes to the discordance, and we hope to be able to segregate that.
Cool, thank you very much. All right, we have 2 minutes left. Let me check. Okay, there is one question for Heike again. So Diamond is very popular. As I said before, I'm a big fan too. So
I'm happy to hear it.
Are Diamond searches additive? For example, if we did a search on 1 billion sequences yesterday and wanted to add another 1 million sequences to the database today, can we combine the searches?
So the short answer is yes and no. But the longer answer is a brilliant question, because what we're trying to do right now is, so the first bottleneck is traversing the tree of life in a quick manner. The second problem is, I didn't have time to talk about, so thank you for that question, is that if you would want to allow unlimited number of hits, not just the top 100 or whatever for one query, we easily scale towards petabytes already with just the NR against the NR itself, for example. We easily scale towards petabytes of output. And so we are trying to now deal with that. So, right, building a database infrastructure that can handle these petabytes of diamond hits. And then, of course, the idea is that not every single person who wants to do a tree of life scale diamond hit should recreate that huge database. Of course, it doesn't make any sense. So we are basically looking into a way to store this huge database and then, of course, try to make it somewhat additive. It's not 100% additive, unfortunately. It would be fantastic. But at least you don't have to recompute anything you already computed in a way. So it's basically some kind of symbiosis.
All right, because this is something similar you can do with BLAST, right?
Exactly.
The problem is that
Exactly.
the database size changes and it's not so accurate, but yeah.
Exactly.
Thank you very much. Thank you. I would like to say thanks to all the speakers for the great talks and questions and answers. I think it's time now for us to wrap it up. Remember that you can go to Spatial. There you can find this in the networking menu on the left side of the live stream. And thanks to everyone for your attention. And we'll see you later. Take care. Bye.
Thank you. Bye.